Welcome back to another past paper review of the Cape Biology Unit 1, Paper 2 from the year 2019. In fact, this is a continuation of this paper. We have already gone through question 1 and question 2, so we're now going to jump into question number 3. Question number 3 is from Module 3, and it is on reproductive biology. The question reads that gametogenesis is the development and production of the male and female germ cells required to form a new individual. The first part of the question states that draw a well-labeled flow diagram of the process of oogenesis starting from the primary oocyte. Now, before I go into the drawing, let me quickly just talk about what is oogenesis. All right, they already established what is gametogenesis. And so I will talk about oogenesis first, and then I go into the diagrams. A matter of fact, for gametogenesis, what it simple is, it's simple the production of gametes. So you're producing gametes, and gametes are sex cells, which includes egg and sperm cells. So once you talk about gametogenesis, you're producing gametes. Let's just make a note of that real quick. So the production of gametes, and gametes will be the same thing as sex cells. All right, and sex cells include sperms and egg. So now let's talk about what is oogenesis first, and then I go into the drawing or answering the question. So this is some extra information here. Talk about oogenesis. All right, and so oogenesis, again, it is the production and development. So let's say the production and development of an egg cell or ovum. All right, so that's what oogenesis is. And if you should say the word spermatogenesis, it means the production and development of a sperm cell. All right, so here we're dealing with the production of egg cells. Now, also what I want to make note of in summary, I want to give you this little piece right here. To undergo oogenesis, or the process of producing an egg, it actually starts from a small cell which is called ooganium. The ooganium is what will produce the cells. I'm just going to write this here. And so ooganium will produce oocyte, and oocyte will eventually lead into ovum. So I just want to give you this flow diagram first. We'll not even start answering the question yet. All I want to give you some information here. Ooganium will turn into oocyte, and oocyte will turn into ovum. Now, what's the difference here? Let me just quickly give you that below this flow chart right here before I get into answering the question. I just wanted you to get an overview of what I'm talking about right here. So, ooganium is those small diploid cells that actually form the ovum. So these are small and they are diploid. So actually, let's just say diploid cell, but just to make a note that they are small diploid cells. So these are diploid cells. And we know that diploid cells are produced from mitosis. Okay, just to make a note. So they are diploid and they produce oocyte, and oocyte, they are immature eggs. So oocyte will be immature eggs. All right. And ovum will be the mature egg. Okay, so just to make you understand that. All right. But now let's go into actually answer the question. And I want to point out that they say start from the primary oocyte. I'm not going to start from the primary oocyte as the question asks. I'm going to go before the primary oocyte. So I'm going to go to the ooganium because that's where I actually start. But if you should answer this exact question, I will show you where to start from. But just in case you may see a question that says start from the ooganium or before the primary oocyte, then you know exactly what to do. All right, so let's start here. Let's go all the way over here. Because I'm also going to label this because it asks you to draw a well-labeled flow diagram. And so, matter of fact, go down a little bit more so I have more space to label. All right, so I'm just going to go right there. Should be good. And so, let me do some drawing real quick and then I'm going to type into this. So the first I'm going to draw is 2N in a cell. So this is going to be 2N, my 2N. And I will label this to say exactly what it is shortly. And then I'm going to take this into another 2N. All right, but I'm going to make this 2N a little bit different. So I'm going to make this here. And I'm going to put a second ring around this one for a particular reason. I'm going to explain that shortly. And then this will take you into an N. And then this will also take you into another N. All right. And also what I want to do, let me change the color of this because I want to draw something else. But I'm going to change this into probably red. Yes, will make sense here. And so what I'm going to do is to make a second, let me use broke, matter of fact, let me contour this bright light line. So I'm going to call this N as well. I will explain all of this, what I'm doing, even by drawing. And then I'm going to break this one again into another N right here. All right. So now let's go into start labeling what is happening here and also to explain. So the first cell I've drawn is the, let me also label this in red. The first cell I've drawn right here, it is the ooganium. Okay, so this is the ooganium. Again, the question did not say start from there, say start from the primary oocyte. But I'm saying just in case you may go in the exam, you may know what to do if they ask you to start before the primary oocyte. And so this 2N will be the primary oocyte. Okay, that's the primary oocyte. And after the primary oocyte, it will go into a secondary oocyte. So you'd have a secondary oocyte. And then you'll go into the last one, which is the ovum. So this one will be the ovum here. All right, so those are the cells that are involved there. Now, what these are, what I put it in red at the side, those are what they call a polar body. But there are two polar bodies. And so here you have the primary polar body. This is the primary polar body. I'm just put it right here. All right, and then you have the secondary polar body. All right. Now, we can also add some more stuff to this. And I just want to make a note. Let me just quickly put this in. I'm going to label this extra layer that I place right here. And this extra layer that I place, it is actually called a follicle. All right. So the oocyte will be in a follicle. So this will be the follicle. All right. So that's a follicle right there. Now, also what I want to make note of is that, let me go a little bit further down. I also want to make note of something specific right here. All right, just to make a note right here is that this cell, let me start from all the way from the ooganium. The ooganium, which I also mentioned before, is diploid. Just to make a note of that, it's a diploid cell, which means it has the full number of chromosomes. And the primary oocyte is also diploid. So if you should explain this and they ask you to explain this, what process of cell division is taking place, then moving from a diploid to produce a diploid, that is mitosis. I'm actually going to label that as well. And then it will eventually go into haploid cells. So these are haploid cells. So we have haploid cells after the primary oocyte. All right, so those are haploid. Again, we can also note that the first cell with the secondary oocyte will produce in meiosis 1, and then ovum by meiosis 2. All right, and so we can also break that up into different sections of meiosis as well. But what I want to label here, and I'm going to put the meiosis part as well into it just to make it clear. And so what I want to point out right here is that these two cells are coming from the ooganium into the primary oocyte that is actually produced by mitosis. And so here, coming all the way down, we're going to have meiosis. All right, so let's just put those in. So this is mitotic division. So this will be mitosis. And going from the diploid into haploid, we have meiosis. Now, the difference here, and I could make a note under meiosis. So let's call this meiosis 1 to produce a secondary oocyte. So this will be our meiosis 1. And by the end of this, going to produce the ovum. So by the end of meiosis 2, that is when you'll have our ovum, which is a mature egg cell. All right, so just to look at the diagram carefully, you'll see it there. All right, so you have the ooganium. From the ooganium, you have the primary oocyte. The primary oocyte turned the secondary oocyte, and then the ovum. Point to note is that the oocyte will be developed into the follicle, and eventually when it matures, it will release from the follicle, which is called ovulation, and the remaining follicle that is left after ovulation will form what they call the yellow body or the corpus luteum. I just point it out real quick. And again, we can also know that the entire process coming from a diploid to haploid is meiosis. And so the primary oocyte will produce by the end of meiosis 1, and then the secondary oocyte will produce by the end of meiosis 2. All right, just to make up, the ovum actually will produce by the end of meiosis 2. So the secondary produced by the end of meiosis 1. The ovum will be produced by meiosis 2. All right, and so the primary oocyte is produced by mitosis. All right, so that is our, I think this is a 3-mark question, I think. Actually, it's 5 marks. Yeah, it's actually 5-mark question. All right, so now jump into the next part of the question. And this one is asking us to state the role of one named hormone in oogenesis. All right, and so I'm going to make it this one really quick, because there are 2 hormones that you really have to focus on when it comes on to oogenesis. And so I'm going to talk about the first one. So they say name one and state the function or role. So I'm going to talk about the 2 of them that are very important in oogenesis. And so I'm going to talk about FSH. All right, so FSH, let me rewrite that. So FSH is actually what they call the follicular stimulating hormone. So this is going to be the follicle stimulating hormone. All right, so follicle stimulating, I missed that, hormone. All right, so that is FSH. What FSH does, let me put it in 2 ways. Let me start right here. So I'm going to put this in like in a 2-point format. And so 1, it aids with the growth of the follicle. So growth and development of the follicle. All right, and it also will aid with the maturing of the primary oocyte. So it also helps with the maturing of the primary oocyte. All right, so very important for that. Also, we have LH. At the end of this, I'm going to outline a few things that you should know as well. And so LH is another one, and LH here is luteinizing hormone. So you're going to be luteinizing hormone. All right, and so here, the luteinizing hormone is very important in the releasing of the secondary oocyte. So this is responsible for releasing the secondary oocyte, and it really release it from release it from the follicle. All right, so just to make a note of that, release it from the follicle. Let's say from the follicle. All right, and just to make a point right here again, which I've mentioned before, that this is actually ovulation process. This is ovulation. All right, so it's responsible for that. All right, so once the oocyte is matured into the ovum, it will release it from the follicle. All right, so that's very important for the LH. Now, what I want to point out right here as well is that FSH and LH, they are produced by the pituitary gland. Okay, so they're produced by the pituitary gland. Now, also what I want you to note is that these hormones that are produced by the pituitary gland, they have the effects in the ovary. So, matter of fact, let me quickly write that down. So FSH and LH are produced by the pituitary gland. All right, and have their effect in the ovary. So you see the effects of these hormones in the ovary, and in the ovaries is where oogenesis take place. Now, these two hormones, they're also responsible in stimulating two other hormones. And so while the follicle is developing and growing, so let's go back a little step up. While the follicle is developing, right, and growing, it will be responsible to produce estrogen. So I'm going to make a note right here that estrogen will be produced. Let me just go back a little bit, one step to change. Let's change the color right there, just to make a notation right here. So FSH, once you produce the follicle, and the follicle is growing, then from the follicle, you'll get estrogen. All right, so just to make a note of that. Now, from LH, once the corpus luteum is formed, or the yellow body is formed, same thing. What you have there is progesterone. All right, so progesterone is formed from the corpus luteum or the yellow body. So they're responsible to produce these two hormones, and so estrogen and progesterone are produced in the ovaries, but they have the effect in the uterus. So let me write that down real quick. All right, so that's another point to note right here, is that estrogen and progesterone are produced in the ovaries, but have the effects in the uterus. All right, so just to make a note of that. All right, so that's something you need to know right there. And just in case you need to explain the effects and where these are produced, they know exactly what to do. And estrogen could spell with the OES at the front, okay? Just to make a note of that as well. All right, jump into the next part of the question. And the next part of the question here is figure 4 shows a diagram of a sperm cell, okay? And so we have 4 labeled part of a sperm cell right there. All right, so let's label quickly before we actually do anything. All right. I know they're going to ask us some questions pertaining to this, but what I want to do is just quickly label, and then I get into the next part of the question in terms of what they're actually asking us. So the A part of this is called a head. So let's label this as a head. All right. And then the C part right here, it is the mid piece or middle piece. Let me just write it as middle piece. Mid or middle piece. All right. D is the tail. The tail is also called the flagellum. All right. So the tail is also called the flagellum. Let me just see if I could put this a little bit. Okay, that's fine. All right. Let me go back into this. I want to go one step down. Great. And then B now, it is not a typically labeled part of the sperm cell for the most part, but you're going to notice that this is called the neck. This is the neck. All right, so awesome. So what I'm going to do also is to give you some other parts that you should know as well. So I'm going to put a little central located piece in the middle right here. I'm going to put a line circle structure right here. I'm going to just draw one, but there are many of them that will be here. A little scribble line. Yep, looking like that. All right, that's not perfect, but anyway, let me draw another bigger one. Maybe you see it a little bit better. So if you see them, you kind of know them. So this is inside is kind of a rough line there. Okay, that a little better now. All right, so what these are, I'm going to talk about what these are, what this is. And so there are many of these in the middle piece here. And then on the top of the head of the sperm, there's something nice there that is responsible to do a proper function that is very important for fertilization. And actually what is present at the top of this, we call it the acrosome. And the acrosome is the enzyme that will be there, that will be needed to burrow through the membrane of the egg cell for fertilization could take place. And this will be the nucleus. And the nucleus is what contains the genetic materials that are necessary to combine with the genetic materials of the ovum to create a diploid zygote. And here we have mitochondria. So this will be a mitochondrion. All right, so that's a mitochondrion. And there are many of them in the middle piece. All right, and so let's get into the questions. All right, so let's jump into questions now real quick. And so the first part is to provide annotations on one function of the following structures labeled A to D in the diagram of a sperm cell shown in figure 4. And so let's go back to A. A is the head, B is the neck, C is the mid piece, and D is the tail. So they're in order, A, B, C, D. And so let's talk about the head. And so the head here, it contains enzymes. So the head is actually a number of functions, right? But it said one function. And so I'm going to kind of mention as much as I possibly can as I go along. But in interest of time, I want to move fast as well. So the first I'm going to talk about in the head is that the head contains enzymes, and the enzymes there, which is acrosome, to contain enzymes that are needed to digest the wall of the ovum, okay, to allow fertilization. All right, so that's very important for it. Not only that, the head also carries, that is in the nucleus, carries genetic materials, so carries genetic materials as well. All right, so genetic materials that are in the head is present in the nucleus. And the genetic materials, which are the chromosomes, and for human beings, it would be 23 chromosomes, that would be necessary to combine with the 23 chromosomes of the ovum to produce a diploid zygote. All right, and so for the B part, which is the neck, and the neck is such as a unique thing. It contains 2 centrioles But two centrioles, they actually needed for two different things, okay? But I'm going to specify one as a concern to the sperm cell itself because it also aids in something to do with the zygote as well. But I want to mention it pertaining to the sperm cell itself because it's talking about the function of the sperm. And so it contains centrioles, so contains centriole. Okay, so contains centriole. As a matter of fact, centrioles, just to make that clear, so contains centrioles that aid with the connection of the flagellum. All right, of the flagellum. All right. So it's very important in terms of helping the flagellum to connect to the sperm itself. All right, because it must have something to anchor to. So it centrally runs through, right through the midpiece all the way down into the flagellum to create that connection. So very important to note that as well. Not only that, the centriole will also help in terms of the first cleavage furrow that is in the zygote. So once the zygote is formed, the centriole that is there will help in terms of cleavage division of the zygote. So let us make sure point that out as well. So there will be a cleavage division that helps with cleavage division of the zygote. And you know why that is important? Because remember, the zygote will eventually divide to make more cells and they form the embryo, because after the zygote you go into the embryo. And so once embryos start forming by cell division, then the central will be very important in terms of the cleavage division of the zygote itself, right? Just to make a note of that. Part C now, which is the mid piece. And so this one, I'm going to say, contains many mitochondria. So mitochondria. All right, so contain many mitochondria to produce energy. And it's producing energy for the flagellum, because the flagellum will need all this energy for swimming. So it produces a lot of energy for the flagellum. All right, and so the flagellum now, which is the last part, it provides movement. It provides movement to enable the sperm to swim. All right, and it is swimming along the female reproductive tract. Very important to note that. Matter of fact, it will swim in the oviduct as well, through the urethra and along the female reproductive tract, just to make a note of that. All right, so very important to provide that energy. So movement for the sperm to swim, and the energy that it gains will come from the middle piece, which contains many mitochondria. All right, so let's go to the next part of the question. And so this part of the question is relate any one structural feature of a sperm cell to its function. So one structural feature. So things you know about the sperm cell that enable it to function well. And so I'm going to talk about the size. And compared to egg cell, sperm cell is really small. So I'm going to make my first point here to be relatively small. All right, and relatively small, if something are smaller, generally smaller objects move faster, right? Because of the less weight to carry around, less energy required to move a larger mass. So the smaller mass require less energy for movement. So generally, this will allow faster movement, okay? So this is for faster movement, just to make a note of that. All right, great. Another point I'm going to make is that the sperm cell itself, it is streamlined. So it is streamlined. All right, so the streamlined Just to make a note of that, is streamlined. And being streamlined, I'm going to put another word here, which is actually torpedo. So it's torpedo shaped or streamlined. But streamlined is the best one to use. A matter of fact, so it's streamlined. And since it's streamlined the way it's shaped, it allows easier passage. Okay? So it allows easy passage. So I'm going to say passage easily through the females, and not just the female, because it goes through the sperm duct as well and through the urethra. But I'm just want to mention it in terms of the female reproductive tract. But again, just for movement generally through any duct, all right, or through the duct, sperm duct, even through the urethra, and through the female reproductive tract. Just to mention that. All right, so passage easily through the female reproductive tract. All right, so very important to note that. Another structure of the sperm that you can also mention, we talked about this before, but I'm going to put it right here as well because it is a structure well featured, contains a flagellum or tail. So it has a tail, right, or flagellum. But since you have talked about that before in the previous part, then there's no need to come back here and talk about it again. You like to do things that you didn't mention before. All right, but if you run out of options and based on number of marks, then why not list them? All right, so if this was like 6 marks, 4 marks, you want to list everything. All right, so this contains flagellum, and flagellum is simple. Allow it to for swimming. So this is just for swimming. So for swimming, let me say for swimming. All right, so very important to note that as well, right? And I could also make another structure that is there that it contain acrosome that contain enzymes, and enzymes allowed to burrow through the walls of the egg, which I also mentioned that before. Okay, so the fact that it contains acrosome, all right, an acrosome again is where the enzymes will help to burrow through the walls of the egg cell. All right, so that is it. But again, the size will be great. The streamlined structure will be great. Contain a flagellum for swimming. Acrosome, where enzymes are created that will help to burrow through the wall of the egg cell. All right, so that will be that for that question there. 2 marks question. All right, so you state the structure and also state the function. That's a 2 mark. All right, so this one now, part B of 1, it says self-incompatibility is determined by multiple alleles as the S locus is at the S locus, a matter of fact, is a complete the following table using the key given below to show the extent to which fertilization is possible between the genotypes of the parents. Now, before I go into answering this question, yeah, let's talk about one simple thing here. When we talk about self-incompatibility, this is referring to hermaphrodite plants, really, right? Or plants that are monoecious. Okay, so monoecious plant or hermaphrodite plants, this is what this question is referring to. You're unable to self-pollinate because you're self-incompatibility. You're self-incompatible. You're incompatible to the self. So the male gamete of the same flower, it is incompatible to the ovule of the same flower. But the truth be told is not necessarily ovule is incompatible too. But the pollen tube will unable to be formed, and hence the sperm cell cannot go into it. Because remember, talk about polar bodies and all of that, right? Because remember, you have the generative cell of the pollen, and you have a tube cell of the pollen. So the tube cell is not able to produce a pollen tube to enable fertilization or allow the sperm to enter the style. Because remember now, the pollen tube is triggered by chemical. And so if the chemicals within the pistil is not compatible with the chemical of the pollen tube, then a pollen tube cannot be formed. And then hence no fertilization. Doesn't make a note of that. All right. So let's now look at the key that they gave us. So we could quickly master this and run through it. So here it said if you use a negative, negative means no fertilization possible, none at all. That means it is totally incompatible. A positive sign means that fertilization possible by all pollen grains. A plus and a minus, or a minus and a plus, means that fertilization only possible by half pollen grains. And so if you think about the word self-incompatibility, mean that you're incompatible to yourself. So to make life easier on us right now to understand this, if the male is the markers on the male is 1 and 2, right? 1 and 2 for the genotype, right? 1 and 2, and 1 and 2 will be the markers for the locus, right? Or the specific type of trait or genes or alleles. Now what I want you to think about, as long as the self is there, then it cannot be compatible. So as long as 1 and 2 is there, it is not compatible. That is self. So always look at what is given and what is the same as the self. So let's call this the self. So looking at this, we're looking at this. Whatever is similar to this, it is not compatible because it is self-incompatibility. So let's look at this right here now. S1 and S2. Same thing as the self, which is the male and the female. So the male and the female are exactly the same. It means that it is not compatible. So therefore, we could put our negative sign right here because in this case, we are saying that this one here, it is not compatible. So let's put a negative sign right there. Negative. All right? All right, let me write that in so it'll be bigger. So you see this negative. All right? And then now, let's put the other one, S1 and S3. Now, when you think about this, if I have a 1 cannot be compatible with the one over here, right? So think about it. So let me just put S1 here to explain exactly what I'm talking about. So you're going to see that a little bit better. So you have S1 and S2 coming from the male. You're going to merge with the S1 and S3 from the female. And so as long as you have an S1, it cannot be compatible with this. All right? Or, matter of fact, I should put it on the other side. So let me just change this around to take out what is not going to be possible. But why I put the X over the male is simply because the male will not able to produce the pollen grain. So actually I'm going to put it back. All right. So again, you'll have your S1. All right, and the S1 is from the male. And so this S1 right here in the female, all right, it cannot be compatible with the male S1. Cannot. So just to make a little marker right here, that this one will cause the incompatibility. Okay, ignore that. Sorry, I don't know why this do like that. Okay, great. I have it correct now. All right, so this here will cause the incompatibility. All right, and so therefore, the S3 is different from S1 and S2. So therefore, you have partial pollination. So the S3 alone will be able to be pollinated by any of these, right? So the S3 can be pollinated, but not S1. All right, so let's put partial where partial is a positive and negative. Yep, only possible by half. So we're going to put a negative and a positive. Okay, let's look at this one now. S3 and S4 is totally different from S1 and S2. So since it's different, it's not the same type of pollen. So the ovule is different from the pollen. Totally different. Right, so it's totally different. So therefore, we have a positive sign right here. A positive sign. So let me point this out again. Hopefully you understand this. All right, let me erase this. You're going to see it a little better. So let's talk about it now. And just very, very simple to make sure we understand this. Anything that is same as the self, let's call it the self, the male gamete, cannot be pollinated because it is incompatible. So 1 and 2 incompatible to 1 and 2. So no pollination takes place here. S1 and S3 is only half incompatible, which is S1. Only half of it is incompatible. So this is incompatible, S1. But S3 it is not incompatible. So therefore it can fertilize by one of these, right? S3 and S4, total pollination because it is totally compatible. All right? And so hope you get that. All right, so now let's jump on to the next question here, our next part of the question. And so here now we said, differentiate between each of the following terms. And so we're gonna differentiate between self-incompatibility and male sterilization. So what's the difference between self-incompatibility and male sterility? Which is the same thing as male sterilization. All right, so let's quickly do that. And then we're going to be out of here real quick. So let me go a little bit down. So self-incompatibility, which we just talked about that just now. So self-incompatibility. All right, so self-incompatibility, it now means, all right, it now means, I'll put a little thing right here. So self-incompatibility means that is the inability, so this is the inability of a pollen, and remember we talked about the pollen earlier, of the pollen to fertilize the ovule. Okay, it is not able to fertilize the ovule. Okay, and let me make sure make a note of this, of the same flower. Okay, of the same flower. So make sure you point it out. Of the same plant. Okay, so of the same flower. All right. While male sterility or male sterilization male sterility or male sterilization is the inability to produce, and since I'm talking about plants, I'm going to say plants right there is the inability to produce pollen. All right, and not just produce pollen, but I'm going to also make a note right here, pollen or sperm, okay? So the sperm cells are not able to produce in the pollen grain. So what I wanted to notice right here now, in self-incompatibility, the pollen grains and the sperm cells within the pollen grains will be produced, but it just cannot fertilize the ovum. But in the male sterility, there's no pollen grain being produced or no sperm cell being produced. And so there's nothing to be fertilized. Okay? All right. And just to make a note that a single pollen grain contains 2 sperm cells. Okay? Just to make a note of that. And you also need to know about your germinative cell and also your tube cell within the pollen grain. All right. And also your polar bodies. Don't forget those type of stuff. Okay? Now let's go into the next part here. That's a 2-mark question right there. And the next one here that we're going to differentiate between is monoece and dioece. Again, these words explain plants that are monoecious and dioecious. And so monoece, think about the word monoece. So mono, it is a single plant. So let's talk about this, read this real quick. All right, so monoece is a single plant or one plant, let's say one plant, all right, having both male and female reproductive organs. Okay, All right, so that's what monoecy is. So the one plant containing both male and female reproductive organs. Now, monoecy refers to hermaphrodite type of plants. Okay. All right. So let's say while dioecy, which is kind of, it can be a little bit confusing if you think about the word di means two. And so you may easily mistake both monoecy and dioecy, because mono means one, which means a single plant containing the two organs. So that's why the mono comes in, one plant. That's why I make sure I say one plant having both. But dioecy now means two different plants or separate plants. So separate as in di, two different, okay? All right, so let's say it as that. While dioecy is when two plants, all right, so we're let's say two different, right? Two different plants, right, have separate, so two different or two separate plants, all right, having two separate plants having a male or a female reproductive organ. And I want to explain this properly to you so you could actually understand exactly what I'm saying, right? So what I'm saying here is that the plants are different, but each of them or one of them containing either a male or a female. So you will not find both male and female on the same plant. There are different plants having them. So you have two different plants, and each of them or one of them having a male, the other one have a female. So the di comes from two separate plants. So monoecy, same plant with both. Di or ecy mean two different plants with either or. All right? So just kind of remember the word. I know it kind of confusing when you hear the word mono, mean only one organ, but no, don't think about it like that. Think about the plant itself. One plant, dioecy, not two organs on the same plant, but two separate plants with individual reproductive organ. All right, so I hope I make that clear, because I know sometimes that can be a little bit tricky in your mind. Think about the words. Now, protandry and protogeny, which we talk about this in paper 1. So I'm going to quickly jump into that, talk about protandry. And this is only dealing with the male and female reproductive parts, which one matured before the other one. That's just what it is, okay? So protandry, this one, protandry refers to, is a state, a protandry is a state where the male, let's say males, reproductive organ develops or mature, okay? The organ develops or mature before the female's reproductive organ. All right. All right. Let's take out the because it's the same flower. So let's take out this, the S's, apostrophe S's. So where the male reproductive organ develops before the female reproductive organ. Okay. All right. And so what I want to point out right here is that there's a reason for this, because this is promoting what they call cross-pollination, because when one matures before the next, it promotes cross-pollination. In other words, the pollen is being produced and ready to be transferred, but the ovules aren't developed yet, so they cannot fertilize itself. Right? So it promotes, so protandry and protogeny actually promotes cross-pollination, right? All right, so while here now, so while protogeny is when the reproductive organ of the female, or the female reproductive, let's say when the female reproductive, it's going to sound better like this, when the female reproductive organ develops before that of the male. All right, so great. All right, so that's the difference between protandry and protogeny. All right, so now let's jump on to the next part of the question here, and this is the last part of the question, is explain two major genetic consequences of each of the following methods of pollination. You know, actually I can only answer one of this because so self-pollination versus cross-pollination, right? Actually I could answer one and that's it. Why I said that? Because if I answer one, the opposite is true for the other one because they're different, right? So the consequence of one is the opposite consequence for the other one. All right, so it's the opposing consequences, I should say, right? So let's say self-pollination. Let's think about it, yeah? Self-pollination, the same flower pollinates itself, which means the gametes are pretty much the same. So the first point I'm going to talk about right here, that there is little to no variation. So here we have little to no variation. Yeah, so little to no variation. Let's kind of explain that. If there's little to no variation, it is simply because the gametes, and this is because the gametes come from the same plant, yeah, come from the same plant. And if the gametes come from the same plant, we can say come from the same plant, hence the alleles, so hence the alleles, let's say the alleles, all right, will be similar or be same. So will be similar or the same. All right. As a matter of fact, there's a high percentage possibility that the alleles will be exactly the same. And I'm gonna give you a word for that because it results in, let me just make a note right here as a second point, as a matter of fact. There's a high chance of what they call homozygosity. So so there is a high level of homozygosity. All right, so the high level of homozygosity, which simply means that the gametes or the genotype will tend to be the exact same thing, which means then there'll be no difference because the 2 points are related. There'll be no difference in the offspring that is be forming. So when they fertilize, the alleles will be pretty much the same. So if the plant is homozygous dominant, it's going to be homozygous dominant. If it's homozygous recessive, it's going to be homozygous recessive. And what happened to, even if a plant is heterozygous, over time, because of continual self-pollination, the heterozygous form will disappear eventually and only leave you with a high level of homozygous type of plant, right? And so that's why self-pollinated plants always tend to produce the same type or same variety or same conditions of the plant. And also what we can also have here again, we have a reducing gene pool. So as I mentioned that over time the heterozygous form will disappear. Okay, so there will be a reduced gene pool. Okay, so reduced gene pool. Not only that may happen too, because sometimes because of a parent plant, what they say now, combining or colliding with the daughter cells and so on, there's a higher chance of mutation, right? Because closer relatives sometimes will lead to mutation, right? All right, so being that they are always cross-pollinating, there's a higher chance of mutation. or not only mutation, but infertile offspring, right? Infertile offspring. So that is also important as well. Not only that, there's a next one I'm going to put right here, is that there's a other possibility that there will be less likelihood of building up resistance. So if there's a disease, so less resistance to disease or to diseases, right? Yep, let's leave it as that. So the truth is now, I don't need to even answer the next part for cross-pollination, because cross-pollination will be the opposite of all of these things. So for cross-pollination, you can say more variation. Why you have more variation? Simply because gametes come from different plants. Okay, I have plant right here. I'm supposed to have plants right here. All right, from the same plant. And so they come from the same different plants for cross-pollination. Hence, the alleles will be generally tend to be different. All right, because if different plants will produce different alleles, because of course the meiosis process will be different in different plants. All right, and so therefore will be a lower level of homozygosity. So therefore you see a greater difference in terms of heterozygous possibility in offspring popping up. The gene pool will be larger because you have a lot of homozygous dominant, a lot of homozygous recessive, a lot of heterozygous. You see a lot of those things. Mutation will be less possible, and the offspring will be more fertile. And also, you can build up resistance to diseases, because a favorite gene from a plant could be transferred to a plant that has a less favored gene or allele. All right, so again, I'm not going to go to this cross-pollination part of it. The opposite in terms of explanation for the self-pollination is true for cross-pollination. All right, so I'm just going to go to the word just for going their sake, but there's no need for that. In interest of time as well, just please rephrase and paraphrase these statements and to make it accurate for cross-pollination. The opposite is true. All right, so here we're going to end this. Again, good luck on your examination. Have a great and blessed term. All right, best wishes.